Okay, I think we're good. Okay. Ah! I'm actually garbage at streaming. I don't know how any of this, like, functions, so bear with me. Uh, howdy. I'm like... Yeah, okay, wait. Is this what I wanted? Yes, this is what I wanted. Okay. I don't know how any of this... Oh, hi. Yeah, well, you can always watch later. Hi. This is, like, incredibly nerd shit, so... have fun. Okay. So hi. This might be a short stream, because I'm almost done with this sheet, and we're kind of out of sequences at the moment. But... yeah, we definitely fly by the seat of our pants here. But yeah, basically, I'm just going through and validating a bunch of sequences. Basically, we do... yeah, so we do our... we use nanopore, so Oxford Nanopore Technologies, to sequence everything. And so basically we sequence like 500 mushrooms at a time, a little less. I don't know what that is. Power by. What is power by? I might, if you... yeah, so we definitely do high throughput sequencing, so it's not as... I guess... I'm not going to say it's not as accurate as, like, Sanger or... yeah, send it. As, like, Sanger or Illumina or stuff, but, oh, I see what you mean. Yeah, our sequencer makes these spreadsheets. It's all on him. I don't actually make these spreadsheets. Actually, I need to... what time is it? 2:28. I'll just remember what time I started. So basically we do like 500 sequences at a time, so we have to kind of go through and make sure that, you know, it is the sequence that goes to the mushroom. There's no contaminant sequences, like there's not something weird going on with it. So there's a lot of like back end kind of work that goes into it. This is obviously INAT. This is the mushroom I'm looking at. It's a little Ascomycete. This is a website called Mycomap. So it's a friend of mine who lives in Indiana made this website, and it's basically like a tool to help like the bioinformatics processing stage. Like, so not really the bio, but after we do the bioinformatics, kind of like the, you know, the let's look at the sequences and make sure they're good. It's so nerdy. So basically what I do is, here's my mushroom. Here's the sequence that I pulled back from mushrooms. So this is like a... nanopore works in multiple reads. So like Sanger is... when you do Sanger sequencing, which is like kind of a much older version of sequencing, Sanger spits out what's called a chromatogram. So it's basically like colored peaks and valleys that kind of show ya, you know, where your nucleotides are. Whereas this is nanopore, and what nanopore spits out is consensus reads. So it basically pulls a little string of DNA that's tagged through pores in the little device that reads it a lot of times, right? And it'll get multiple reads of those strings of DNA. And what it'll spit out is a file like this after the bioinformatics is done, that is just an aggregate of different reads. So every one of these is an individual read of this gene sequence. And then when I align it, which is up here, it is very cool. Yeah, that's how it was described to me. Like basically there's like a little tag sequence that gets attached to the beginning, like a little tag primer that's attached to the beginning and the end, and it uses that to read it. Yeah, it's crazy. Like you literally have this tiny little drop of once you've done all the processing, um, there's like this tiny little drop of liquid that they'll put in the nanopore. And sorry, I'm looking for something. I have a plate. Where? I have boxes everywhere. Ah, here we go. Okay, so like basically we have like these are the plates we use for sequencing. So each one of these each one of these plate wells is a fungi sample, and then we'll do like 5 to 10 of these a run depending. Yeah. Versus Sanger where it's like one at a time. And you probably can't even see it. Like the samples are absolutely it's not even going to focus. Like you can't, the size of the samples we take are so tiny. Like they fit on the head of a pin. You probably can't even see it. Itty bitty bitty bitty. Because if you overload the DNA, it'll kill the pores on the machine faster than we want it. Yeah. I literally just have boxes of mushrooms over here that I have to get around to sampling, but I've been too distraught to do anything about recently. Okay. Anyway. Yeah, like if there's too much DNA, it'll totally clog the pores. So yeah. So basically, yeah, the distraught's not good. So we have our little aggregate file, and usually, at least our sequencer for here, so these are fundus sequences. So our sequencer will do a forward primer and then a reverse primer. So half of these reads are in the forward direction, and half of these reads are in the reverse direction. And that motif I was mentioning earlier can be used to kind of figure out where your forward read is. So I type it in, it finds the forward read, and then I can go and select the other. I got nerd shit. And it like takes forever to line because like it just does. So Eugene is an open source program. There's a more expensive program that's a little more powerful called Genius. It's expensive, and this works fine. So I just use this. Let's see. So basically what I'm doing here is I'm just looking at this, I'm looking at this sequence because when it, so NG species ID is kind of the thing that, yeah, Blender's fine. God, it's been so many years since I used Maya or Blender that like I couldn't even tell you. it's been over a decade since I touched either program. What was I saying? Yeah. Okay. So when it spits out my consensus read here, it will not spit out ambiguous nucleotides. So any spot where like there's 2 or more haplotypes and one haplotype might have a T in the position, another haplotype has a Y in that or a C in that position, like you would put a Y there because it could be either, but my program won't spit it out. So I usually go and try to find the ambiguous nucleotides. And I kind of have a trick of doing this. I'm sure some scientists, like academics somewhere, would probably tell me I'm wrong, but whatever. This one probably isn't going to have any ambiguities. But yeah, this one does not have. Oh, like, of what do you mean? Like getting tricks to work around the free programs and make them do what you want them to do? So I'm not going to edit this sequence at all. Possibly, yeah, send it to me. I can send it to him. He might have heard of it already. The guy is an insane workhorse. Yeah, and actually Micromap is really cool. So the way Steven set this up is it'll pull all the data from the NCBI database, so GenBank, which is the US government kind of like DNA repository, but also it pulls stuff from Unite and Unite I can't my brain. Unite Bold. It'll pull stuff from Danny Miller's annotation. So Danny is a huge uh not huge, but like very researched West Coast mycologist. Like this is his website. And honestly, if you ever have since you're in Washington, if you ever have questions about mushrooms, you can totally go look at his website here and you might actually like it might help you figure some stuff out because he's got really detailed notes on a lot of different stuff. Um It'll pull stuff from Mycoportal and it'll pull stuff from like iNaturalist and Mushroom Observer. So like we can compare stuff not against just NCBI blasts, but we can also compare stuff to like stuff in the local database, which helps because a lot of the stuff in NCBI isn't well curated. Right? It looks like shit, but it's functional. So, like for this mushroom, I'm looking at it, and my big thing I'm looking at is this identity number. With fungi, depending on the genus, we're mainly looking at ITS 1, 5.8s, and ITS 2 for barcoding, which is what all we're doing. We're just doing barcoding, right? This is not whole genome sequencing. In ITS, variability between species can be a single base pair, or it can be like up to 4 or 5% depending on the genus. So there's a lot of like wiggle room. Um, yeah, it's really like it really depends. Like stuff like Hebelomas and Cortinarius, and like some Armillaria. There's a couple genuses that are really hard to differentiate with ITS. Yeah, but again, this is just like one small gene region, right? This is only like a 400. It's just a very highly conserved section. Like in a lot of groups, especially the basidiomycetes, it's a pretty highly conserved region. It doesn't change that much. Um, but there are some older groups that do have a lot of variability, so it's a little bit like this one. Yeah, this one, the closest matches are only coming out about 98% similar. Like my closest one is 98.36 here. That has decent query cover. There's a 98.69. They're all under 1% similar. Usually if there's a 1% difference is when I probably will assign it a temp code. And like when I'm assigning temp codes to things, I'm not saying this is for sure a new species. It's just like a helpful convention to like group similar sequences together so that if someone does go like write a paper on something, they can say, okay, this group and this group, and then they can go do a tree and do the micro and do everything and decide, okay, these are separate species, or these might just be the same species and these temp codes need to be combined. So I'm actually probably going to put a temp code on this one because there's not really any matches above 99%. Yeah, it's like data processing versus like actually describing species. Like I've seen a few people get like weird about like, well, why'd you put a temp code on this? And it's like, well, because there's no good reference data in the database and I'm just looking at sequences, so I can't say that this is for sure this species because no one's done the micro work and there's not a type specimen sequence and yeah, exactly. A lot of what we're doing is just trying to get the databases worked up because there's none. So I'm gonna put a temp code on this. This is Definitely Pizziza though. Yeah, of course. I was in a good habit of streaming like before the election, and I've just been in a very bad headspace the last couple weeks, so it's not been great. Um, but our goal, I mean, like, there's kind of like three labs in the US right now that I'm aware of kind of doing this like general biodiversity survey stuff that we're doing. Yeah, basically, me too. Um, one's in California, which is Fundus. So I work for Fundus. There's one in Ohio, which is the Ohio Mushroom DNA Lab, and we also a 501c3, but we haven't managed to secure a decent grant yet, so everything we're doing is volunteer with OMDL. And then there's my Coda Labs, which is in Indiana, Michigan area, and we've probably produced between the three labs, we've produced more sequences than. Yeah, I'm glad I get the California money too, because like grants are, whew, the grant market is not great. Um, but yeah, we've between the three labs, we've probably produced more sequences than uh, just anybody else. Like tens of thousands of sequences, and a lot of it is just, at least with OMDL, like a lot of OMDL is community funded, which is pretty good. Like basically, we've been doing free sequences, and people are kind enough to send us money when they have the means to do so. So we're trying. Like it requires a special kind of nerd, but we are definitely trying. We've been, and like a lot of this, part of the reason I've been like trying to do like streaming and stuff is just so if we have people that want to like go through and validate their own sequences, because this stuff is becoming a lot more accessible. You can get a nanopore if you have a couple grand to drop. You can get a nanopore machine. Like it's the min ions are only a couple $1,000s. They're actually approachable for like a layman to purchase if they really wanted to. Yeah. I mean, I think it just depends on how well funded the clubs are. I really wish we had science equivalents. The problem with a lot of the mycology clubs is some of them are getting a little more on board with the really science side of things, but a lot of people are in it for foraging edibles or like psychedelics, and that's fine, but eh. It's just hard to convince a local club to drop thousands and thousands of dollars on sequencing equipment if they really just want to like figure out what bolete is tasty. Yeah. Yeah, Washington's good for like both food and y'all have a lot of good psychedelics. Like I'm not like a huge psilocybin person at all. Like I have friends are, but I'm like whatever. But um mm? Oh, psilocybe? Yeah. Yeah. But they're still freaking cool, right? But like Ohio does not have a lot of psilocybe species. There's only three that I know of, really. So there's Psilocybe ovoidiocystidiata, which is mostly found like in the Ohio River Valley. It tends to associate with like floodplains and box elder. There is Psilocybe, oh my gosh, Cerulean rhiza. which OMDL put out a paper. We actually just described that this year. Like our sequencer for OMDL. Yeah, right here. So yeah, like where is it? Yeah, this is the holotype of Psilocybe ceruleariza right here. I think this is the holotype. Yeah, holotype. Just found in a mulch bed near Cincinnati. These fruit in the winter. OMDL put out a paper on them a couple months ago. And then we have Psilocybe cerulipes, which is the only one I found of the three. It's just tiny and it's teeny itty bitty and tends to grow with hemlock. Right, because you're into paleontology. But like, this is why INAT's so flippin' cool, man. Like, we have full color photos, very detailed full color photographs of the holotype specimen with DNA sequence information attached. Like, that's so cool. Right? It is IRL Pokemon Snap. Also, this is why it's called Ceruleariza, because the rhizomorphs, so the mycelium, does this. Like, there's actually bluing in the rhizomes that it makes. How flippin' cool is that? Of course, Alan took this picture. Yeah, Ceruleo rhiza. Yeah. Like, it's so freaking cool. Yeah. What was I doing? I was validating this thing. Oh god, like when they're named after a person. The pound Dunkleosteus. Yeah, I mean, there's definitely some mushrooms that are named after people. It happens. But I do the descriptive ones are nice. Please stop collecting rusts. But there's also some like absurdly long fungi names, like Ovoidiocystidiata. Say that 3 times fast. I'm actually a super weirdo, and I made a list on here of mushroom names that are just too fucking long. Like what were the like Marasmiellomycena pseudoomphaliformis. Fomus. What? Callistosporium purpureum marginatum. Like, it's just and then what happens is people like mush together like prefixes and suffixes, and this has happened. Like, so like Rhizome marasmius. So it's a marasmius that has some like rhizomorphic I'm assuming it's because of the stem. So rhizo marasmius instead of just like. But oh my god, it gets too much sometimes. It's just funny. And then you get like species epithets that get used like multiple times that are like really common. And then you'll have a genus that is like almost the same as another genus, and that happens sometimes. So there's Gymnophilus dryophilus and Gymnopus dryophilus. The scientist did not. Mega. Oh god, yeah. Megacolibia is a big one. Oh, what is this mushroom? There are very few. Let it cola, let it cola. Yeah. I'd say mega's not as common in mycology just because there's not that many huge mushrooms. Uh, but it does happen occasionally. Okay, so this is one of those weird flippin' cases where all of my reference data is from bold. Like, so I've got a lot of bold matches and I've got a lot of matches in GenBank, and I always start getting suspicious. Fair. The only one I can think of is the genus Megacolibia off the top of my head, which isn't really too bad. All things considered. But yeah, I always get suspicious of GenBank stuff because there is a preponderance of uh GenBank stuff that hasn't been well curated. And so you'll have like these dump taxa where like they'll just say, "Oh, well this is a super common genus name, obviously." And then there's like a 10% spread in what the actual ITS is, and they're definitely not the same species. Yeah, you always have to be a little bit like, I'm not going to say sus with GenBank, but like take a second, who put out the sequence? Like, is it an actual expert in the genus, or is it a college paper from a plant pathogen person that doesn't actually give a shit what the mushroom, like, because a lot of these will be like environmental samples, and they're not even like pulling these samples from like a macro fruit body. They just like from root tip testing or something. Oh, wait a sec. That's only 17 RIC. Hmm. CS. CS? Like, I should know what CS is. I don't understand the abbreviation, I'm sorry. Oh, Citizen China. Yeah. Yeah. And like my goal when I'm going into this is try to be as thorough as possible, but also be as like, yeah, computer science, yeah, that makes sense. As thorough as possible, but like also as conservative as possible, if that makes sense. Like I don't want to make unnecessary changes, but I want to like try to keep stuff as, I don't know, I want to avoid problems like perpetuating in the system, because like if someone starts calling something by the wrong name and it perpetuates, it's a lot harder to fix it down the line. Oh, well, this is interesting. Why, yes, I care about the truth. Okay, this is definitely that. Let it cola. Wait a second. Justin's wrong. It is an astoundingly low bar. Like, I sometimes feel like I'm living in crazy land because, like, even in, like, the mushroom community, there are a lot of people that are, like I said, they're not really there for the science. They're just kind of there for the vibes. And so there's a lot of questionable information that gets thrown around that is, maybe not entirely accurate. Like, some people, if you hang out in foraging groups, you'll see this. You'll see people like picking up mushrooms with gloves because they're afraid like if they touch them they'll get hurt. And these same people will like with their entire chest like yell at people for touching the mushrooms or eating the mushrooms or whatever. It's like, but like you're not proclaiming yourself to be an expert. Outside of allergic reactions, there aren't any mushrooms that I am aware of that will hurt you by touching them. Fungal toxins they just don't work that way. Plants are more dangerous than fungi. Yeah. There's like one Japanese coral mushroom that kind of had some like, oh, it's the Japanese fire coral, it'll hurt you if you touch it, but it yeah, there's there, like some people have some allergic reactions sometimes to like some of the slimy mushrooms, but no, touch them. We are more comfortable with plants, but there's a lot more plants that you can brush up against and they will make your life miserable. Miserable. Like you can even take a bite of the most toxic mushrooms and as long as you spit it out, you're fine. Like I could go find a destroying angel, take a bite, chew it up and spit it. I'd be good. No harm. I've heard like one anecdotal story of a mycologist getting a little bit of mild contact poisoning from some amanita, and she was working with some extremely juicy ones and got like a lot like on her skin, and it like I mean, I usually if I do a spit and taste on a mushroom, I usually like rinse it out, but realistically to actually get amatoxin poisoning, you have to ingest probably a healthy adult would probably have to ingest at least like a cap or so. Like even if a couple tiny pieces, like it's not gonna. They're toxic, but they're not like okay. They're very toxic, but you still have to digest it. Yeah, and there are some groups of mushrooms where like tasting them is an important diagnostic. Like if it's in anything in Russulaceae or Boletaceae, if I'm like taking notes, I'll usually taste it because whether or not they're bitter or spicy is like a big identifier. What am I doing? This is Chris. Yeah, people. Well, the problem is America is just very mycophobic. Like, there's not a lot of our initial colonization came from like Europe, not Europe, but like England, Britain, like British culture, and British culture is mycophobic compared to a lot of other European countries. And so like we just inherited that mycophobia. Yeah. And honestly, there's way less toxic mushrooms than like I feel completely safe foraging for mushrooms. Completely safe. Um Not that I've heard of, but I'm not going to say it's not a thing. There the one instance of like a legit mycologist that I heard of that died from eating mushrooms was this guy that I want to say yeah. So this guy Julius Schaefer Paxillus. So he died from eating Paxillus. I believe it causes rhabdomyolysis or something. I can't. I would have to look into it. Um, I can't remember the exact story, but it might have been because this guy spent so many years eating these that it like built up in his system a bit. But don't quote me on that. I would have to like double check, and I just don't have time of that. Yeah, dig into that rabbit hole if you want. But so far, I really haven't heard of any like mycologists becoming sensitive. Like you have to remember, like mostly we're looking at fruit bodies. We're not really looking at so with plants, like plants have a lot more defense mechanisms because they're trying to stop us animals from eating them. Mushrooms are ephemeral, like they're a fruit. So, and honestly, some fungi use being eaten as like a dispersal method for their spores. So, I just, it's, yeah, that's probably just it's one of these things I could probably dig into more. I don't know. I don't know. I prefer to say I don't know. They are fruit. I've definitely been in the woods before and seen squirrels chowing down on Russula. Could probably. People. Yeah, except sometimes people find um God in the mushrooms. I don't know way better to say. Yeah, okay. Here's a squirrel munching on a mushroom. Look at him go. He's just vibing. He doesn't give a shit about me. He's just like, I have a mushroom. Yes, the look at the trees people. No, I definitely know a few people who are like, God's in everything. You find unscientific thinking in every group, and when you get a bunch of people that take psychedelics commonly without actually having any scientific grounding, it can get real woo real fast. Oh yeah, it's yep, exactly same type of woo. Which is fine. I'm not gonna yuck anyone's yum. They're not really hurting me. But yeah. It's kinda oh shit, I forgot to check something. Yeah, the woo I'm more tolerant of the woo most times because like most people who are into it aren't really pushy about. Most people aren't that pushy about. Some people are pushy about it, but I can't tell. Oh no! Oh no! No! I mean, like, if you want to just get healthier by having smoothies, sure, but also medicine is good. Yeah. Oh, the raw milk people are sending me. Like it's the regulations around milk came from the blood and lives of so many dead children. Like it's I, especially with a bird flu pandemic that is potentially crossing the barrier into mammal transmission, I will not be drinking any raw milk any fucking time soon. Yeah. Vaccines and pasteurization and all of these are built on the backs of so many lives. And the fact that people just refuse to acknowledge it, it's fucking just, it's wild to me. So, why are these all so low RIC? Yeah. It's just infuriating. Like, it's like we should not be having issues with things like, I don't know, fucking polio. Like, holy shit, if smallpox was an issue in this day and age, people would just, we would not fucking eradicate smallpox. Fucking unbelievable. It's wild to me. Absolutely wild. I did that wrong. Oh, yeah. Oh, yeah. You know, something's gonna unfreeze in that Russian permafrost and just kill us all. Yep, not concerning in the slightest. But hey, it's okay. Oh, that's weird. It, yeah, I don't know. All I can do is just try to do what I can, and maybe my small contribution to the world will help someone in the future. That's, I think, my copium at this point. Yeah, I'm a little jealous of the West Coast. Ohio is, chewy bit concerning. Just discuss. Alright, this is probably gonna be. Yeah, Texas is worse than Ohio. Definitely. Like, Ohio is, I mean, I keep saying Ohio isn't fucking gone, but we voted out Sherrod Brown, so I don't even know if I can say that anymore. Oh yeah, my husband used to live in Seattle. Washington's great. Okay. Okay, not doing that one. But I don't know. Hoping I can stick it out here. Oh god, Florida. Yeah, nope. I barely want to visit Florida. No problem. Yeah, I should probably. I might pop out soon too, because I am definitely getting distracted, and also I'll stay on for a bit. Um, at some point, I'm gonna have to like get off and do some annoying upload stuff. Yeah, glad to help. Enjoy the nerd talk. Oh, let's see. Stereo. Yeah. It's like hard to talk and work at the same time. Like I'm okay at multitasking, but there becomes a point. I'll give it maybe like 20 minutes more, then maybe pop off so I can, because I gotta do some like real just obnoxious tedious stuff today. That's not gonna, no one is going to want to watch me do. That's not what I wanted to do. I hope you're learning something about fungi here. They're fun guys. So bird's nest fungi are fun. So these guys, these are like the spore packets, and so when raindrops hit them, it kind of pings them out of like the little cup and like shoots the spores out. Super weird little dudes. But they're like they're usually super tiny, like itty bitty little guys. I mean, actually, yeah, you can see this is this is a centimeter, and they're about 5 millimeters wide. Some of these I went through and already edited the sequences on, because it usually takes it takes map a couple minutes to do the new blast search. Like this is called a blast. So for my sanity, I usually go through and do the edits and then come back to them, so I don't have to like sit there and wait for the system to finish processing. Those are pretty common. Okay. So remember when I was saying earlier that some mushrooms are only like 1 or 2 base pairs apart? This is one of them. And actually, I'm gonna guess it's gonna be one I can't get to species. Yeah, so Cortinarius croceus and Cortinarius tertii are only about a single base pair off, and we have found a couple so far that when you adjust for ambiguous nucleotides, like you have a multi-haplotype situation, there are some that are coming out after you adjust for the ambiguity is matching both the holotype for Cortinarius croceus and the holotype for Cortinarius tertii, which is interesting. It's definitely an interesting problem to have. um, we're not sure if it's a hybrid situation, if the two species are actually conspecific and need to be combined. I don't know. I don't know, but it's definitely something we've been noticing. Um, let me find it. Yeah, we basically have this project going that's like all the ones we found so far that are questionable. Like the haplotypes are different enough that we're like, what's going on here? Again, it's not any case of me trying to be like, this is for sure a hybrid. It's just something's up. Someone should look into it. So, but yeah, so this is this matches the holotype sequence for tertii, and this is the holotype for Cortinarius croceus. And so when I align them with my sequence here, this is not the right sequence though. I don't know what I'm doing. We're just gonna get rid of that. We're just gonna do some shit here. We're gonna copy that. So top one's tertii, second one's croceus, third one is my edited sequence. Yeah, different enough, yet similar. So what we can see happening here is, so right here, ATW. So W is the ambiguity that represents A and T. And then we have AG, and R is the ambiguity code that represents A and G. And then we have AAM, so that's kind of interesting. And we have AAW. But really, it's these two, that one. These are the two base pairs that are normally different in Crucius and tertii. And this is not the first one we found where it has both haplotypes. So, what we've been doing with these is sticking them at section, which is below genus, but above species. So they are in section Dermosibi. But yeah, they are. Like, this is the nice thing about nanopore sequencing. You can actually kind of pull out and see the individual haplotypes, which is great. You couldn't really do that with older styles of sequencing, but it does kind of lead you to rabbit holes. I've been down some rabbit holes as a result. Mm-hmm. Oh, I've fallen into some rabbit holes so deep before that I had to just like clock out for the evening and walk away from my computer and think about it. Like, it's eh, like I have to stop myself sometimes from getting a little too into the weeds. I'll add that later. 100%. Yay, I'm glad you like it. This is like kind of high level stuff, so I'm happy that you are enjoying this weirdness. Like this is a level of science nerd that it's just not common. Let's see. Solid ideas. Subgenus dermosity. I also wish that niche science nerdiness was more common. I've managed to mostly populate my YouTube feed with that sort of content, thankfully, but it's it definitely takes some effort to curate because it just keeps trying to get me to look at stuff that isn't that. Uh, this is honestly kind of like why I got into watching Erica's channel. Like she'll sometimes just like go through scientific papers, and I love that shit. Like at some point, I'm gonna get real bored and just like start going through some mycology papers, like especially like if if we publish some over the winter. Um, yeah, my YouTube feed is very politics oriented right now, unfortunately, and like I for my own mental health, I probably need to move it away from that, but I can't for reasons. Oh, I don't know. It's just hard. Oh, hopefully Forrest will put out some more sciencey videos soon. I can hyper focus on those for a bit instead of like doom scrolling politics 24/7. All right, what do we have? We have some bunch of from completely random matches. Oh god. Okay. Uh, mhm. Honestly, there's a couple. The Eastern North American species of Macrolepiota were recently described, and the paper is open sourced, and I'm actually very tempted to go. I might do that. I might like go through the paper on those because yeah, it's gonna be hard to get away from it. So this Macrolepiota species and this one were recently described by this guy, Steve. Um, and like we've known these were distinct species from the European species for quite a while, but it just took a long time for anybody to describe them because there's just not a lot of people putting out. Oh yeah, they're not uncommon and they're a very good edible. Um, but there's just like not a lot of people putting out papers. Oh, someone cleaned that up. I bet it was Alan. So there's just not a lot of people putting out fungi papers, so it took forever to get them described. And then there's like another species out here on the west coast that is distinct that still needs described that we have like sequence data on. But literally when those two species were published, I went through INAT and got the entire continental US cleaned out of the improperly identified species, because Procera is not present in the US. We have not a single one has come back being Procera sensu stricto, as like in the European sense, in the strictest sense. But this one's weird. I don't know what's going on with this one. Hmm. Ricus. Okay. There's a type sequence somewhere. Where's the type sequence? Fucking Christ. This is my problem. Like, someone will say, oh, there's a type sequence, but the type sequence isn't fucking uploaded to GenBank, and then it's like, where am I looking? What am I looking for? Furnace. Be careful. I know I said Macrolepiota is edible, but be careful you have your ID solid, because there is a very common lookalike called Chlorophyllum molybdetes that will it'll make you projectile vomit. Don't eat those without significant amounts of processing. Hmm. Well, if you ever do get into it, feel free to run stuff by me. If you're in Illinois or anywhere in the Midwest, like I'm going to be familiar with those species, like of edibles. Hmm. I can't brain this one right now. We're just gonna look at a different one. I need to do some digging to find that type sequence. And actually, yeah, that sucks. There are some. I mean, look for like state parks. But yeah, the plain states aren't exactly the best for fungi, unfortunately. Um, they just really aren't. It's just too arid. I mean, though, you can find some like cool. There are still fungi. They're just going to be different fungi. I mean, I've found a mushroom in the desert before. They just look weird. Yep, that's that. Hmm. Your best bet is to look either like during a really nice, like solid multi-hour rain, like the little like ephemeral stuff will start like the little marasmuids will start popping up on like the grasses and stuff, or like a couple days after a good rain is when like I'll notice the big macrofungi kind of popping up. Um, You just gotta find like the microclimates they like. Like fields tend to be really good for like giant puffballs and stuff, so that's at least like a good edible. Mhm. But yeah, I mean, moisture is really what you're looking for, unfortunately. Um, moisture in forests. Like a lot of mushrooms are mycorrhizal, like so they have associations with like plants and especially trees and such. Um, so like old growth forests will tend to have a lot more species diversity than something that's been like tilled and replanted because there's just some of those species get lost when agriculture comes through. Like there's nothing to really replenish them. There are some very easy mushrooms to grow in like your garden bed that are good edibles. Look into wine caps, Stropharia rugosanulata, and bluets, which are Calibia nuda. Uh, wine caps tend to fruit in the summer, and bluets tend to fruit in like fall, winter. Like I have a patch in our garden, and our particular the genetics of our particular patch tends to fruit like really late in the fall, but like some of them will fruit a little earlier. Like low temps seem to trigger the fruiting on those. Um, we had a good rain, and it's getting colder, so I suspect our patch will start fruiting soon. But you can probably find sawdust spawn of either. Like North Spore is kind of like the easiest company, but they're not the cheapest. But there are other companies that do make spawn for them. And if you get like sawdust spawn or something, you basically just like mix it into your wood chips and leaf litter, and they'll start popping up. It's basically just like already inoculated uh, straw or sawdust, and you just mix it in. Um, I mean, you could always. There's also like stuff like oysters and I mean, there's various species you can grow. But I mean, if you're talking like edibles you can grow in your wood chips, those two are the ones I would probably suggest. The other ones you kind of need a slightly different setup. Yeah, totally do it. And the nice thing about inoculating like your garden beds with like fungi is it'll help kind of break down the wood chips and the leaf litter a little faster, which is like good for the soil health and good for the plant health. It's just good, right? Alright, I'm gonna do this last sequence and then probably pop off because I gotta do some annoying stuff. Like I've got these sequences up here that have been hanging on that are gonna require some obnoxious and intense editing that's gonna result in me staring at the screen like a crazy person, and no one wants to watch that. Uh, Basically. Yes. Like, there are like gardening practices you can do where you like integrate vegetables and edible crops into like other more beneficial plants for the ecosystem, and it's just fucking good. Like, I planted a bunch of Helianthus tuberosus sunchokes, which are mm, theoretically native, but at the very least naturalized. They've been cultivated as a fruit crop in this country for so long that no one's really sure like where they come from. Um, mm, that's not quite correct. The info I have found is muddy, right? But the tubers are edible and they're like a great pollinator plant. So like I planted a bunch and I now have a bunch of tubers and if we're ever in an emergency situation, I just have food in the ground that I can go dig up. Like, and like we have blackberries and we have currants and we have uh we're gonna plant some other veg this year and hopefully, work on we're trying to like just integrate things. But we also want to have a garden that's like good for pollinators and good for insects and good for birds. Like, we have a ridiculous amount of fireflies in our backyard every summer. And the reason we have a ridiculous amount of fireflies in a backyard every summer is because we don't get rid of our leaves. Like, I literally just like push them into my garden beds and just let them live there. We barely do any garden cleanup in the fall. Like, I usually wait until it gets warm in the spring to do a lot of like heavy trimming. And so I have stuff like miner bees and carpenter bees uh leaf cutter bees uh lots and lots of fireflies. But if you look at my neighborhood, like if you go 4 houses down to the guy that obsessively mows everything, and God, heaven help you if there's a single leaf left in his front yard, like no fireflies are in that guy's yard. It's just silly. Yeah, I don't use pesticides. At most, the most I will occasionally use is maybe a little neem oil, and even that I've gotten away from. Dude, my neighbor across the street is one of those guys that will mow his yard at like 8 a.m. on a Saturday and then use a gas leaf blower for this tiny postage stamp of a yard for like an hour. He is so obnoxious. It's it's completely weird. I don't it's I get it. Some people have yards as their hobbies, but holy shit. I couldn't do it. It's crazy. Like, the guy's yard looks good, but it also looks lifeless. Like, oh, dude, it's people. There's leaf pickup around. People like push their leaves to the curb, and like big trucks come around and just pull all the leaves out of the neighborhoods. It's like this is why the insect population is going down. Meanwhile, I'm planting like native food plants. Like, I planted some pawpaws this year, and I'm super excited to get fruit out of them. But like also they're hosts for a lot of insects. So like it's food for me and also benefiting the ecosystem. Who'da thunk? Yeah, same. I just I don't know, I think there is a way for us to live with nature and not fight against it. Yes, do pawpaws. You have try to get wild types if you can. If you can find some own root wild types, that would be ideal. You have to have you can't have clones. You have to get multiple genetically different individuals because pawpaws are not self-fertile. So just keep that in mind. I planted two bare roots or like own root ones. They're not like grafts or anything. And then I have a neighbor that's like a block away that also has some pretty mature pawpaw trees. So that's also going to be a vector for pollination for my guys. But yeah, with pawpaws, you have to just be aware they will not pollinate themselves. Pawpaws are great though. They're like so good. Warning though, they're better raw. Like we've tried doing like pawpaw pancakes and stuff, and they are fantastic. But something about cooking pawpaws makes them actually harder on your digestive tract than if you eat them raw. And so some people get really bad GI distress from eating cooked pawpaw, and I am unfortunately one of them. So they are great in smoothies though. Pawpaws in the wild, they tend to like river bottoms. Yeah, they are very similar to banana. They're kind of like banana yogurt custardy ish to me. Um, yeah. I'm lucky. Our neighbor down the street who has the pawpaw trees, like he has way more crop than he can ever eat. So he just like sits the extra fruit down at the end of his driveway. He's like, "Here, take some." It's very sweet of him. They are a very sweet couple. Like that's the kind of shit I want to do. Like if I have enough fruit, if I have extra fruit, I would totally just share it with people because that's like I don't know, that's like fucking anarchy, baby. That's like community minded ethics. It's like helping your fucking community and sharing food. I'm all for it. I actually have a bunch of yeah, exactly. Like we don't put out extra like berries because it's really easy to freeze berries, but like with the pawpaws, it's just like whatever, eat them. They're only ripe for a couple weeks. Might as well enjoy them and spread the love. But yeah, if you're trying to find them in the wild, you really got to look for like river bottoms. And the unfortunate part is if you find like a colony, it doesn't even mean you're going to find fruit because it might just be a clonal colony. And if it's all one genetic individual, they might not pollinate. Yeah, tomatoes can get way ahead of you if you're not careful. Actually, one of my favorite like plants to tell people about. Let me look it up. So it is the Genius Impatiens. Touch me nuts. So these guys, Impatiens capensis and Impatiens pallida, these are very common in like wet areas, right? Like they're like seeps or river bottoms. So the leaves of these guys. Let's see if I can get a good picture of the leaves. That's a better picture. So the leaves of these guys, if you are in the woods and if you run into like poison ivy or nettles or something that's like a skin irritant, you can actually like take the leaves off of one of these and kind of rub them against the irritated spot and it will soothe your skin. So like if ever and like it's nice because they grow with nettles. So if you ever run into a patch of nettles, if you can just find some jewelweed, you can just rub it on the sting and it'll go away. It's like so very, very convenient because I usually find the nettles. Oh, this one's weird. What is happening? Okay. Oh. Hmm. Yes. It's a super useful plant, and like not everyone knows that. I actually worked with the a botanist at our local metro parks this summer, and he thought he had heard that, but he thought it didn't work because he had been like trying to squeeze the juice out of the stem and then like take the juice and rub it on. Ah, yes. One sec. Let's see. So there is a book that I don't have. It's called Appalachian Mushrooms. Let me pull it up. It's by Walt Sturgeon. Appalachian. I need to buy this one. This one is a good one. I don't have this one yet. I still need to pick it up. But yeah, leaves, not stem. So this is a really good one. Walt is kind of an old school mycologist in the area. He's not really like a genetic guy, but he is very knowledgeable and a very good guy, and he knows his stuff. So this is a good book. Anything by Michael Kuo, so like this, anything by Michael Kuo is going to be a good book. He's who runs mushroomexpert.com. I'm just pulling books out. Wow. You wouldn't be a science nerd if you didn't just have a pile of books. Oh, here's another one. Okay. So yeah, Appalachian Mushrooms is a good book. I actually really like this book. This is Mushrooms of West Virginia and the Central Appalachians by William C. Rudy. It's a little out of date, so some of the names aren't going to be correct, but it's just got a decent kind of collections of mushrooms, and like the pictures are decent. It's not like bad pictures. It's just a nice, well put out guide. This is a nice little field guide. It's not very big, but it's actually small enough you could totally bring it into the field with you. And he does detail like some edibles. This is Wild Mushrooms of Pennsylvania and the Mid-Atlantic. This is another. Yeah, I can I can just type you let's see. I'll hit you up in the Discord. I'll hit you up in the Discord. But Mushrooms of the Northeast. This is by also by Walt Sturgeon. There's also one that is Mushrooms of the Upper Midwestern Peninsula, I think it's called, which I don't. Yeah, Upper Midwest. So same kind of publisher. I don't have this one, but it's also going to be like a decent little field guide. Like some of these books aren't great if you are looking for like the most up-to-date scientific names, but they're a good like starting point. This, The Complete Mushroom Hunter, is what I usually suggest people who want to start with forageables. Like this is not an advanced book, but it does kind of go over like what bad ones you need to look out for, what ones are good edibles. Like it goes over basically the easiest edibles and also the shit that'll kill you. So like that's like what you need to know. This is the Quo book. This is just a generally really good Amanita book. So this is Amanitas of North America. There's also a really good website that's amanitaceae.org, but it's kind of technical and hard to get through. But like this is like the Amanita Bible. Yeah, don't eat the stuff to kill you. Yeah, good Amanita book. Amanitas of North America by Britt Bunyard and Jay Justice. And then this isn't the only Bolete book by the Bassettes, but this is one of the Bolete books by the Bassettes, and basically the Bassettes are the Bolete people. Any book by the Bassettes is great for Boletes. Like again, this one's like out of date on the scientific names, but I end up referencing this a lot. And this one's fairly recent. So this is the most recent edition of the Audubon Society Mushroom Guide, and it's not like specifically for like the East and the Midwest. Like this covers the whole country. But I know the guy who helped edit this, and he's extremely knowledgeable. Some of the common names are a bit weird, but like the info is good, if that makes sense. I have more books, but that's a good start. Like I said, I'll list them in the Discord later once I'm done. I have so many books. That's literally, it's like half the mushroom books I own. It's a problem. Um, this is what species is this? Pezzaloma? Yeah, and yeah, and the problem with like mushroom common names is just like there's a lot that just don't have common names, but what the Pezzaloma? But there are also just I don't know, like the common names just tend to be a lot more fuzzy. Like I can say destroying angel, but that's going to basically refer to any mushroom in Amanita sexumphalloideae that's a white mushroom. Like if you say death cap, people usually assume that's Amanita phalloideae, but if you say a destroying angel, there's like 10 different species of mushrooms that could be. But if you say Amanita sexumphalloideae, all of those are going to kill you, so don't eat those. Okay, so those are Azaloma? That is a genus that isn't even in... No, there we go. Oh, it isn't genus. Sweet. Hmm. Oh no, they'll melt your liver. Like, so amatoxins, so if you eat them, you'll feel like a little bit sick, and then you'll feel fine for like a day or two, and then you die. Like they are cell destroying toxins. like like they're not just GI toxins. Like they will destroy your liver. Like if you don't catch it and get treated, it's basically liver transplant time. Yeah. Yeah. Don't eat those. Don't mistakenly eat those. um I like my liver too. But honestly, most of the recommended edible mushrooms are recommended edibles because it's very hard to mistake them for something that's deadly toxic. At most, they might have like GI irritants as lookalikes, but even with a lot of the GI irritants, if you cook them enough, it'll render them fairly safe. That's interesting. Like, probably the biggest misidentification that I see is jack-o'-lantern mushrooms, Omphalotus illudens. They're like a big orange mushroom that grows from dead wood in a cluster. People will mistake them for chanterelles because chanterelles kind of are superficially similar, and so people will like get their Omphalotus, thinking it's a chanterelle, eat it, and then end up praying to the porcelain throne for a day. But they won't kill you; they're just going to make you feel miserable. And honestly, if you're paying any bit of attention, you should never mistake Omphalotus illudens for a chanterelle. Chanterelle don't have true gills. Like they have the hymenium. So hymenium is the pore surface, like the fertile surface of a mushroom. So the hymenium on chanterelles, Cantharellus, uh, they're not really gills. It's just like folded. It's like little folds that kind of look like gills, but they're not gills. So you can't really like peel them cleanly off the mushroom like you can like a true gill. Um, and they're not knife edge and they'll kind of crisscross. So it's like really easy to differentiate them from not a chanterelle. It just requires paying attention, which some people are bad at. This is another group 1 intron. Why do I keep getting so many group 1 introns? Oh yeah. So, yeah. If you hang out in the mushroom ID groups on Facebook, there are some wild takes. Absolutely wild. Like some of the groups are good. Like the Ohio group's actually pretty heavily moderated and is decent. Some of the groups are awful, and I don't understand why people feel comfortable just confidently giving incorrect information that could get someone killed. But here we are. Some people are just not like they don't treat this stuff with the sort of gravitas that it should be treated, in my opinion. That's just my opinion, though. It's my problem. I run my mouth and I have opinions. People don't like when you have opinions. Yeah, people are flawed. Mhm. Nope, yep, it's 100%. 100% that is the case. Um like some people just they hear something or they've learned something from like someone down the road and they take it as truth. Like there's a lot of rumors about testing for mushroom edibility. Like you'll hear people say like if it tarnishes a silver spoon, it's a poisonous mushroom. It's like that's nonsense. Or if it bruises blue, it's poisonous. It's like, no, not really. Like there is an old army manual, like emergency guide for testing if plants, like unknown plants are edible, where like you rub it on your skin and see if you have a reaction, then you like touch it to your tongue and see if you have a reaction, then you take a little nibble, like you do like progressively more intense tests to see like if this is a thing you can eat. But like that's really meant for survival situations, and I have full ass seen people parrot that for mushroom tests, like mushroom identification, and like everyone's like, no, that's absolutely not what you have to do to identify if a mushroom is edible or not. Please stop suggesting that to people because it is objectively incorrect and you're going to get someone killed. Yeah. Like you can rub an Amanita sexuifloideate against your skin, but like you're not going to have a reaction. But if you eat it and wait to see if you have a reaction, you might die because there's no like progressive effects. It's like supposedly a mushroom that tastes delicious. Like people have eaten these and said they taste good. Right, but there's also a lot of people that haven't been trained how to distinguish like good sources of information from like complete bunk. And AI is making it worse because if you go to Google, a lot of the top results are going to be Google results. Like here, let's just like AI, like edible mushroom Ohio. And you're going to get this fucking AI overview. And some people like legit will do this and then post it in like these ID groups. And it's like some of this is okay information, but like this just says bolete mushrooms. There are toxic boletes. You do not want to do this. Like if someone just said, "Oh, edible mushrooms," and looked at this AI result, they might go poison themselves. This will get someone poisoned. Also, avoid mushrooms that are moist or slimy or no. Like false morels are generally gyrometra, and there are plenty of edible gyrometra. And also, morels can kill you if you eat them raw. You need to cook morels to eat them. Like it's not good. It's objectively not good. So that's my soapbox on why I hate AI when it comes to mushrooms. And like these aren't even things that are hard to learn how to ID. Like this is not none of the species of mushrooms that like in our area is considered a good edible is um gonna require microscopy to figure out. You every single like choice edible mushroom is completely identifiable based off of macro characteristics. Yeah. Yeah, Google is yeah, not I don't know. Eventually AI might reach a point where I'm not like just mad at it all the time, but it's definitely not at that point yet. All right. Anyway, I'm gonna pop off. Nice chatting with you. I will do this more once once I have more sequences to look at. I need to do some of these off camera because they're just gonna be a huge pain in the ass. Um, yeah. Thanks for watching. Yeah, they hallucinate a lot. I've managed to get ChatGPT to make up fake article titles attached to a real DOI, like completely unrelated article within like 3 queries. Like legit just entirely got it to just make shit up. And it only took like 3 questions. Wild. Anyway, I'm getting off my soapbox. Everyone have a great day. Later.